Dr. Wessler is a distinguished professor of genetics at UC Riverside, and that's where she and her team investigate transposable elements and their role in genome evolution. The title of today's talk is Transposable Element Mediated Structural Variation from McClintock to Pangenomes. Dr. Wessler, can you come up here, please?
Good morning. It's great to be here, and I really want to thank the organizers. I'm going to start in the past because that's where the study of genomes started, and I think Ari described the sequencing projects. What I'm going to go is before that to the person I think is a real pioneer. In the talk today, actually, I'm going to start. This is it. We're going to focus first on McClintock. Barbara McClintock, who I had the great fortune to know at the beginning of my career, with my postdoctoral project beginning with her strains. But she was fundamental to many of the processes that we have discussed, especially relating to genome instability. And to go back to the very beginning, when it was recognized. Okay, I'm pressing, there we go, okay. So this first paper in 1931 with Harriet Creighton was the first paper that recognized that chromosome breakage, well, we know that recombination is the fundamental process on this planet for generating diversity in genomes. McClintock was the first to recognize that recombination involved the breakage of chromosomes. And she did this because she was a master of the technology of cytogenetics, which was really the way that people looked at genomes way back then. And she was the best in the world. She's a cytogeneticist. And she integrated that with genetics and was able to show in this paper by visualizing chromosomes and by visualizing the knobs at the end of chromosomes that the movement of the knob to a different chromosome coincided with a new phenotype, the phenotype that was linked to it. In this case, the waxy phenotype, which I'll be talking about just a little bit at the beginning of the talk. My postdoc project, as I said, when I was in Nina Fedorov's lab way back when, also began with Barbara's material. In this case, it was, again, the waxy gene, which we had to isolate. And then from that, we isolated the AC transposable element and the DS element. Well, McClintock had isolated this material, and genetically, what she concluded was that DS was a deletion derivative of AC. And sure enough, we found that when we cloned the element from the AC allele, from that strain, we had a 4.3 kb insertion in the waxy gene. When we cloned the DS derivative that she had isolated, there was a 200 base pair deletion in that AC element. So, as predicted by McClintock, the DS element was a defective AC element missing this 200 base pairs, which basically turned an autonomous element, AC, into a non-autonomous element, DS. Another place where McClintock has had fundamental contributions, perhaps the first, is towards what she called changes in phase. And again, this was work that I was involved in with Steve Delaporta when he was at Cold Spring Harbor, and with Paul Comet, who's now, I think, retired from Monsanto. And so what we did is we looked at what's called a change in phase, again, material she had isolated. This is at the waxy gene, where we had an AC element, well, she had an AC element that went from active to inactive. And she was able to see this because of the kernel phenotypes, which were so expressive. And you see it, okay, so I touched something and it made it smaller. It's okay. All right. Thank you, okay. Afraid to touch anything now. So what you could see in this kernel is that only a sector of the kernel has activity, has a movement of transposons. So what Paul Comet and myself and Steve Delaporta did was to isolate these genes. And essentially what we showed back in 1987 was the inactivation of maize, of the transposable element, correlated with DNA modification, with its methylation. So in the inactive state, the AC element was methylated. In the active state, it was demethylated. So these changes in state involve reversible methylation, which we now are very familiar with. It's epigenetic regulation. So again, and at my favorite quote, McClintock's quote, came at the very end of her Nobel lecture in 1984, where she said, in the future, attention undoubtedly will be centered on the genome, with greater appreciation of its significance as a highly sensitive organ of the cell that monitors genome activities, corrects common errors, senses unusual and unexpected events, and responds to them often by restructuring the genome. So, you know, people talk about the people who are special in their lives influence you and whatever, but in this case, not only is it somebody that I, you know, fortunately knew and have tremendous respect for, but the material that she provided really propelled my career and the career of so many other people in genetics and plant genetics. All right, so I'm actually going to come back to her because she had another impact, but I'm going to start with our story. This story started about 20 years ago with the discovery in my lab of mites, a type of transposable element. So this story started with actually the first paper in my lab, which was with Rita Varagona in 1985. And in that paper, we had the waxy gene isolated because we used it to isolate the AC and DS elements. But so there was a huge collection of spontaneous mutations of waxy isolated by Oliver Nelson at the University of Wisconsin. And he characterized these using recombination, one of the first eukaryotic recombination maps. And so he identified all these mutations. And the reason these mutations of waxy existed, because when waxy is a starch biosynthetic enzyme, it encodes a starch biosynthetic enzyme. And when it's mutant, you end up with cornstarch. So there are lots and lots of agronomically important mutations. Oliver collected it, made this map, and we went ahead, Rita and I, back in 1985, and did what you did back then to characterize these, which was southern blot analysis. Okay, remember that? For some of you. And what we found essentially is that the majority of these mutations were insertions. And they were insertions, many of them of retrotransposons. And my lab went on to study these retrotransposons, but one of them opened a door that led to the project I'm going to talk about for the last 20 years. And that was this insertion called B2. And B2 was a stable mutation. It was a small insertion of about 125 base pairs. And Tom Bureau, when he was a postdoc in my lab, he's now a professor at McGill, what he did is he isolated this, he sequenced it because we had the gene. And this was when PCR had just come out in 1990, 91, and he was able to use PCR because the waxy gene had been sequenced to go in and sequence this. Before that, we had to make lambda libraries. In order to clone a particular allele, we had to make lambda libraries of the entire maize genome, which is the same size as the human genome. So we had, you know, these trays and trays, cafeteria trays of phage that we would plate out and do, you know, take your filter paper, and it was a mess. So PCR was fabulous, as you all know, but it was really special for us. So we were able in a couple of days to isolate this B2. And what Tom found, again, in the beginnings of doing genomic analysis, at this time, people were starting to sequence genes, in this case, plant genes, and send the sequence to these databases. This is before GenBank. And so there were all of these plant genes that people had contributed, exons and introns, into this small database. Tom used the B2 sequence, scanned that database, and what he found is that many of these genes had distantly related little transposable elements in them, in their 5' flanking regions, in their introns, and in their 3' flanking regions. So this led to our discovery of what is now called mites, miniature inverted repeat transposable elements, that we now know are the most abundant transposons associated with plant genes. So much like ALU and line sequences in mammalian genomes. So let's move on. What are mites? So transposon families are, they have members, autonomous elements, which encode the transposase. I told you before about AC. They have non-autonomous members, which don't move unless they're in the presence of an autonomous element. And mites are, in fact, non-autonomous elements that have the ability, unlike other deletion derivatives elements, to attain very, very high copy numbers, meaning hundreds and even thousands of copies, which is very unusual for DNA transposons. The elements that usually attain high copy number are the retrotransposons. So these are mites, and they're they're not only common in plant genomes, they're also common in zebrafish and C. elegans, and there are ones in humans also. So mites, as I said, are non-autonomous class 2 elements. They amplify essentially from a single element to very high copy numbers in the hundreds of thousands. Where do they fit in what I call the genome ecosystem? And this is something that actually was discovered, the fact that maize genes, and for many plant genes are, as they're called, genes sitting in a sea of transposable elements. And this is what Jeff Benetton discovered years ago when he looked at a yak, when he sequenced a yak around the ADH gene, that there were just 100 kb of retrotransposons between genes. So mites fit in that they are small elements that insert into and around genes, into plant exons, into plant introns, and 5' and 3' flanking regions. So the problem with the analysis back in the early 90s is that all we were looking at were existing elements that didn't move. We were looking at alleles that had insertions in them. So as I said, mites are the most abundant type of plant genes. But for me, beginning with McClintock's active elements, to really learn about how these elements work and how they impact genome evolution, we needed an active element. So if you take a mite sequence and you blast it against, a particular sequence of a mite in an organism, and you blast the genome, what you generally get is this star phylogeny. And what that indicates is that sometime in the distant past, the element attained very, very high copy number, and then activity stopped. And once the activity stops, the sequence is then senesce. And that's what you're seeing here. So what we wanted was to catch a mite in this red circle area when it was bursting, because it's at that time that you could see the mutations that it is capable of producing, sort of the raw material of the diversity that we're seeing in the genome. So Ning Jiang, when she was a postdoc in my lab back in the early 2000s, almost 20 years ago, the rice genome sequence had just come out in 2002. So working with Sean Eddy's lab at WashU, what they did is they used a software package called Recon, and essentially took the whole rice genome and compared it to itself and looked for identical sequences that look like mites. So when a transposon is bursting, when it's brand new, all the sequences are identical. And it's at that point that's what we were looking for. That's what Ning was looking for. And she found in the rice genome, and we didn't realize how lucky we were to find this single element, 430 base pairs. In the Nippon-Bare sequence genome, there were about 50 copies. 40 of them were identical. Okay, so she found this element Mping. Fortunately, the rice genome was sequenced the old-fashioned way with like most reference genomes, with BACs and YACs. And so it was a complete sequence. So in the Nippon-Bare genome, we were able to find one copy of the presumed autonomous element, which we call Ping. So Ping is the autonomous element. It has to be present for Mping to move, and it's the transposase that's driving this burst. And I'm probably not going to talk about Ping a whole lot. I'm going to talk about this burst. Okay. So we had 5 strains, 5 accessions, rice accessions, 4 rice accessions, where we knew Mping was present in very high copy numbers. We got these, and I don't have time to go into it, but we got these 4 strains from a Japanese collaborator. It was Tanasaka's lab, and the collaborator was Yutaka Okamoto. So what he had 4 strains where this element was moving, and high copy number, like 500 copies of Mping. So the reference was 50. These strains had about 500. What we wanted to know is was the element still moving around? And we did a technique called Transposon display, which was also developed in my lab. And essentially what you see here are 3 generations of rice seed. The first panel is F1. Those are individuals taken off the plant. The last individual there on your right, the F0, I'm sorry, was then taken. It was planted. So this is just direct, and then you take a seed from the last one you planted, and you could see each of the bands represents a different element. And you could see that the element is increasing in copy number right before our eyes. Okay, so, and you could sort of see in that last, like if you take the last lane and you go forward, you could see that the new bands are segregating, which is exactly what you expect because the new insertions in the plant are heterozygous. Okay, so essentially what we found is that there were about 10 to 40 new insertions per plant per generation, which was just, we couldn't believe it. The question first, so how widespread is the burst? So this burst was occurring. Where was it occurring in rice? We were able to address that question recently because of the 3000 Rice Genome Project. And Jinfen Chen, when he was a postdoc in my lab, looked at these 3000 sequences. The good thing is that if we're looking at Mping copy number, we can use Illumina reads because it's only a 400 base pair element. We can find insertion sites. Illumina reads are useful. So what he basically found, and you see it here, is that all of the rice strains shown on your left, that's just a tree of phylogenetic tree of the relationship of these 3000 strains. All of them have Mping that you see in the middle, but low copy number, like 20, 30 copies of Mping, because Mping is in the wild progenitor of domesticated rice, of all domesticated rice. But essentially, the only place we found the burst was in the 4 strains accessions that we had, plus a few other accessions that are related to those. So this is very, very, very new. Like we've tracked back the burst. It probably started about 100 years ago. So the only similar situation that we know of, and we're really hoping now that genomes are being sequenced at incredible numbers, that other bursts will be detected. The only example that I know of is in Drosophila, the P elements, which have just, rammed through all populations of melanogaster, and they're now going through simulins. So that's another situation where you can look at the impact of transposable elements in those early stages. Okay, so this is important. I'm going to talk about a successful element. And what do I mean by a successful element? So most transposons are unsuccessful, meaning they can't attain high copy numbers. And the reason they can't attain high copy numbers is because they're mutagenic. And when they're mutagenic, they cause lots of mutations, they're silenced, and so they're usually present in genomes in high copy numbers. Those are not the elements that, for the most part, contribute to genome evolution in a significant way. Successful elements, as I said, attain high copy number because they possess these biochemical features. There's something about them, I'm going to tell you about that facilitate their amplification. I call these their superpowers. Okay, so what are the superpowers of Mping? So one is it massively amplifies through the genome into like up to 1,000 copies that we've seen so far without seriously impacting, without killing the host. How does it do that? It actually does that in an incredibly simple way, which surprised us. Mping avoids exon insertions. It's not impossible. There are some exon insertions, but it has an AT-rich target site, a 9 base pair site that the transposase prefers, where Mping can insert into, and it's generally AT-rich. Rice exons are GC-rich. So it avoids exons in rice. We see some, but it generally, we see non-genic region insertions because of this, feature of its host. Okay, so what I say here is successful amplification reflects a unique interaction between the Ping-Mping biochemistry, that is the AT-rich target, and the right genome features. And this is something I never expected. We've taken this element, and this was done by Wayne Parrott, Nathan Hancock, when he was in Wayne Parrott's lab, took the element, put it into soybean. Soybean has AT-rich or not GC-rich exons, and it's much more mutagenic in soybean. So it's unlikely that this element would have succeeded in soybean. Okay, so another superpower is Mping copies in the genome, all the copies, whether we look at the 50 copies in Nippon Bare or the 500 copies in these high copy strains, they're all methylated. So the host recognizes, and these are the methylated sequences. This is work done by Lulu and Jinfeng Chen when they were postdocs in my lab. All copies are highly methylated, meaning there are small RNAs all over the place that feed back and lead to the methylation of the DNA Mping sequences, okay? And that's because Mping, if you think about it, Mping is all over the genome. It's in different orientations. It's in introns. It's in places that get transcribed. And so we have small RNAs from Mping all over. This feeds back to the DNA and methylates all Mping sequences. But as I mentioned before, Mping is derived from the ends of ping. Ping produces a transposase. So when you look at ping sequences, the only sequences that are methylated are its ends. Its coding regions, the two genes that encode together encode the transposase, are not methylated. So it's almost like Mping is going under the epigenetic silencing radar. It's recognized, but it doesn't matter. It doesn't turn back and shut off Mping. It doesn't turn off the transposase, sorry. Okay, so as it says here, as the burst continues, the host recognition of Mping doesn't matter. Ping continues producing transposase. Its coding regions remain unmethylated. Mping increases in copy number, diversifying the genome. The burst spread to a population, I say, generating significant and unique classes of diversity, which I'm going to talk about in the remaining time frame. It says, let's look at how it spreads to a population. Okay. The population that we chose to look at, I've gotten from Yutaka Okamoto's recombinant inbred line population. So in this case, what Yutaka did was to take one of those high copy strains. I'm afraid to touch my trackpad here. Take one of the high copy number strains, which is this HEG4, which has 7 pings. That's the other thing. The bursting strains have more pings, more transposase. There's more amplification of Mping. Okay, it had about 500 Mpings. Cross it with the reference genome. You get the F1, and then you have 275 RILs. So what we wanted to look at were phenotypes. We wanted to correlate Mping insertions with phenotypes, but it turns out we spent many years doing this. And it turns out there's not much phenotypic diversity because Mping is not causing, at least at this level of Mping copies, it's not impacting gene expression to a significant extent. It is tweaking things, and I'm not going to go into that. But it's part of its success. It's not that mutagenic. So what we decided to look at is the genomes of these lines. Where is Mping going and what is it doing in these 275 RIL lines? Okay, so all of them were sequenced using Illumina short read, and the positions of all the pings and Mpings were determined. All right, so the first thing we found was there were over 14,000 new Mping insertions in the 275 lines, okay? We found only 17 new ping insertions, which I'm not going to really talk about. So but basically all of the RILs that have new Mpings, and they can, some of the RILs had like 100 new Mpings. All of the RILs that have new Mpings have ping. So there's no silencing. We found in the entire population, nothing was silenced. If you had a ping, you had new Mping insertions. And we have 14,000 of them. And I'm going to talk about those at the very end just a little bit. All right, so it's spreading through the population without triggering silencing, at least at this level. Okay, so what we decided after we looked at those insertions, we were interested in structural variation. And that's the part I'm going to spend the rest of this talk on. So I'm going to first talk, so for years and years, you look in the literature, everybody sort of is assuming, not assuming, associating transposable elements and structural variation. So when you see the structural variations in pangenomes, you generally see this association with transposable elements. All right. So first, I'm just as a refresher, although this I'm talking to the preaching to the choir here. Pangenomes are now what people are really focusing on in terms of diversity in populations. And you have your copy number variation, you have your presence absence variation, you have inversions, you have large deletions, etc. So we've come to appreciate that there is a core genome containing the genes that the entire population or species, probably the entire population have. And then these dispensable genome, which are really often the adaptation genes, okay? And as we all know, one reference genome is not enough. Okay, so as I said before, there are more and more papers coming out which are talking about these adaptation genes that are differentially present in a population, but they're associated with transposable elements. And we see this everywhere, as I said before. You're seeing transposable elements are there. And what people generally say is, okay, you have these regions of recombination, and there's recombination that occurs, but recombination is, you need breakage. I mean, recombination doesn't just happen. It has to be catalyzed in a sense. So in one of my favorite examples is, and as more genomes come out, in fungal phytopathogens, this beautiful study showed that you have all these accessory chromosomes that are dispensable, present in different members of a population. And it's those chromosomes that are riddled with transposons, whereas the core genome has far fewer transposons. Again, this association at a level that's, just really impressive. So back to McClintock again. All right. So again, the idea that, as I said before, recombination is enough. You have to actually break the chromosomes. You need a cut in the chromosome to initiate recombination. So what McClintock found, and this was the first thing she found, was that transposable elements break chromosomes. So you go back to her original studies, and DS, her discovery of DS, dissociation. Why is it called dissociation? Because there's chromosome breakage. And this is at DS. So this is the phenotype that she studied. The phenotype was this patch of colorless allurone on a background of wild type purple. And she was able to show, again, the way she showed recombination and chromosome knobs was she set up her chromosomes just right so that she was able to show that there was a site of chromosome breakage where you lost these markers. And she called that site DS. But the important thing about that site is you only saw this phenotype. You only saw breakage when AC, the autonomous element, was in the genome. Without AC, you got all purple kernels, but with AC, someplace else in the genome, you got breakage. Okay, so breakage only occurred when AC was in the genome. Now, the important thing also is most DS elements don't break chromosomes. We know that, she knew that, and it was followed up with molecular analysis. And what makes these breakage chromosomes special was shown by us back in '93, and it was shown by others. It was shown by Hans-Peter Doring and Peter Sarlinger's lab, and it was shown by Jonathan Jones at John Innes. And what Cliff Weil, whom some of you may know, he's an NSF program director now. When he was a postdoc in my lab back in '93, he was looking at a waxy allele where you had a DS and the DS would transpose, and DS transposes locally. It transposes to nearby sites. So we get these double elements. And in fact, when we got those double elements, we got breakage. Cliff was able to show using PCR that the breakage was due to aberrant transposition. So when you have two elements, essentially DS, like many other transposes to local sites. These double elements promote chromosome breakage by aberrant transposition events. So you essentially get the two elements, and when you're getting replication, you're getting mitosis or meiosis. You'll get transposase bound to the end of one element and bound to the end of the other, bring it together and get some sort of structural variation, breakage. Okay, so the question we asked, and we asked this a long time ago, was does Mping transpose like DS and like many other transposons, to local sites? Does it transpose locally? And the way we addressed that question was to take a tDNA with the Ping transposase and Mping, put it on tDNA, and look in Arabidopsis for where it was transposing. And in fact, it was transposing all over the place. So Mping does not transpose to local sites. It transposes randomly around the genome. Okay. Mping copies found throughout the genome, no preference for local transposition. So we basically, this is one of its superpowers. A transposon that transposes to local sites and causes breakage. Breakage is highly mutagenic. So Mping is transposing all over the place, rarely causing breakage. And so that's why we call it a superpower, because it allows it to be more successful. Double Mpings are rare. They're rare, but they occur. So think about this. As you increase in copy number, the chance that you're going to have one element inserting next to another increases just by random chance. And so we had all of these RILs. We had essentially many chances. So what I asked Jinfen Cheng to do was to look at the RILs, the 275 RILs, and look for SV, structural variation. And he found several of interest. So he found all the structural variations that he was able to see, not all that were there, with short reads. And essentially, more than half have Mping at the breakpoints. So essentially what he found was, and we have others that don't have Mping at the breakpoints, but more clearly Mping was increasing the frequency of structural variation. This is an example, a couple of quick examples of what they look like. The gene you see here is in, the chromosome you see there is in Nippon-Bare, the reference genome. The two Mpings are inserted in one of the high copy strains in HEG4. When we looked through all of the RILs, we found one RIL that had a deletion of the entire region, where the Mpings had served as a megatransposon, removing this. So this is one example of a type of structural variation. Another one was a similar thing. Nippon-Bare had this gene. The Mpings in the high copy strain HEG4, the other parent, were inserted in an intron 5' flanking region in one of the RILs. So out of 275 RILs, one of the RILs had this deletion of that region. Okay, so that our data suggests that the Ping-Mping burst generates structural variation rapidly and at a high frequency, and by the following mechanism, and it's just a verbal mechanism. So essentially, structural variations are generated during the ping, during the burst, when transposase catalyzes two different events. One is it catalyzes the high copy numbers that are necessary to generate these double elements, two elements near each other. The second thing is it generates the transposase, the active cuts that occur at the end of the elements, leading to aberrant transposition. And it's this aberrant transposition that's been shown to be due to double elements leading to structural variation. And what we predict is that it'll lead to copy number variants, presence, absence, and inversions. We've only seen the presence absence variation because we've been looking at Illumina reads. Okay, so only structural variants, presence absence will be detected. Okay, what we're doing now, we're involved in nanopore sequencing and others that we just talked about before. So we do have one significant inversion that was detected in the parent lines, because the parent lines we sequence completely. And that's an inversion of 135 kb. It's two of the accessions, EG4 and HEG4, which had a common ancestor about 30 generations ago. And what we see is that there were two insertions of Mping and HEG4 that led to an inversion, 135 kb inversion involving about 10 genes with Mping at the ends. So here we know that it's possible, so we know it's worth looking for. And so the future analysis, Yutaka Okamoto's lab at Kyoto University has two more real populations that they provided us with. And these are even more exciting because with the first real population, we had the reference line, Nippon-Bare, and a high copy strain. Here we have two populations that the parents are both high copy. So this should significantly increase the number of insertions and structural variants. So these RILs have probably well over 1,000 Mpings and are going to have, 10 to 15 or 20 pings, much more activity. Okay. So RILs will be sequenced, and we're sequencing. So what we're doing is we're doing Illumina sequencing on the 500 of the two populations, looking for all the ping and Mping insertions. Then we're going to pick the ones with the most insertions and do nanopore sequencing to get hopefully all of the large structural variant events. And I want to end with a project that's underway using these populations that we're doing with Bob Schmidt's lab at the University of Georgia. And this addresses the question of the impact on the epigenome. So we have these three populations, okay? And as I said before, we've done Illumina on the first population, the one on the left. We found, as I mentioned, 14,000 Mping insertions, new insertions. These are germinal events that are occurring in the 275 RIL. So these are stable mutations that we could study. We have two more populations. We estimate that each will have about 20,000 Mping insertions. So we're going to have, over 50,000 Mping insertions. What Bob's lab is interested in is the ACRs, accessible chromatin regions. And he's interested in, these are the regions of open chromatin that are presumed to be important for regulatory function. And these are very important because in genomes like maize, where you have, hundreds of kb upstream of a gene, the question is, where do you look for the regulatory regions, distal regulatory elements? And the positions of ACRs, of accessible chromatin regions, are, essentially they're associated with many of the QTLs. So they're thought to be the regions of the embedded regulatory regions. But the question is, there's lots of ACRs. How do we test whether they really are regulatory? And as I said here, but incredibly, Bob's lab, Haidong Yao in his lab, has found that almost 50% of Mping insertions are into known ACRs that he has found in the Nippon-Mari, the reference genome. So what this is a population of mutagens, of potential mutagens, to see how many, so we have strains now. An individual RIL may have an insertion into an ACR. When you do RNA-seq, you'll be able to see whether or not the transcription of the nearby gene is impacted by that insertion, whether or not. And this is going to be, this is a new NSF grant we have. This is going to be a community resource of these, because he's using single cell technology here. We're going to look at the, first he's making an atlas using Nippon-Mari, a single cell atlas of all the ACRs and all the cell types, et cetera. And then we're going to start looking at some of these Mping insertions, and they'll be available to the community. So as I said here, I'll end by saying transposable elements shape up otherwise conservative genomes in ways that we are just beginning to understand. And I would thank my collaborators, and I think there's actually a few minutes for questions. Thank you.
So if anyone has any questions, run the mic over to you.
Okay.
I'd probably just get it started with a question about, do you have any idea why the ping doesn't get silenced? So specifically the transposase. And
Good question. So the question is, why doesn't ping get silenced? Ping will get silenced if it goes into the right place. The first thing is it doesn't, okay. In those populations of the 14,000 new Mping insertions, I said we only have 17 new ping insertions. Okay, the way ping will get silenced, if it inserts into a place in the chromosome where you would generate antisense RNA, okay? It hasn't, and that you need a lot of insertions presumably for that to happen. Mping, it turns out, transposes better than ping. So there's a base pair change that's occurred in the right next to the terminal inverted repeats. And we've done we've published this. Mping is a better substrate for transposase. So its copy numbers will increase preferentially to ping. And as I said, at some point, ping and some of these strains will shut down. But we haven't seen it yet. Yeah, it's cool. Okay.
That's really interesting work.
Thank
I'm
you.
really curious about the intron specificity that some of these ping variants have.
I'm missing your question,
oh,
sorry.
Sorry, the intron specificity
intron.
that the ping has. Do you see a lot of splicing variants associated with that?
Okay, do we see splicing variation? It's interesting because the answer is we haven't looked, quite honestly. I mean, we haven't looked. One of the reasons mites are successful in plant genomes is because plants have generally small introns. So, unlike in mammalian systems where the average intron size, I think, is 2 kb, the average plant intron is about 200 base pairs. And we think it's one of the reasons that, mites are successful, can insert into plant genes. But, in mammals, you have these line elements, 2, 3 kb that nicely sit in an intron and don't affect splicing or don't impact it too much. The first discovery of Mping, it was actually discovered, but not recognized. Like 2 or 3 years before our study, like in 1997, where a Japanese group had looked at a mutation in the constance gene of rice. So it's a temporal photoperiod. And the mutation had an insertion in it. And that insertion was Mping. And they never did anything with it. We found it after we discovered Mping computationally. And the Japanese labs had discovered it with Mping mutants. So it does cause mutation. But it rarely does that, right?
Fascinating, thanks.
Thank you. Okay.
I think I might have missed this, but do you know what the signals are that trigger transposition? And for these heritable mutations, do you think that that's triggered? Can you tell what developmental stage it's triggered in?
Okay. So what triggers transposition is the production of transposase. So Ping produces transposase. We think plants don't set aside a germline like animal cells. So if the ancient, not ancient, the revered maize geneticists of old showed very clearly, like Marcus Rhodes showed this, that if you have a mutation in a pigment gene and you can follow a red stripe that goes right through the plant into the tassel, and it ends up being heritable, and a heritable excision. So wherever transposase is made, we see the movement of transposons. The question of developmentally. So Bob Schmidt's lab is able to use single cell. So he could look at somatic mutations. He could look at new insertions that happen. And so if you take a whole leaf and he does his single cell magic, and they can identify cell type by the marker of open accessible chromosome. So we're now looking with these lines at, is there a cell type where more Mpings are inserted, looking at the somatic insertion. So that's the way he's addressing that, but we don't have data yet. Right. Okay.
All right, I've got one last question for you here from online. 1 second. What other superpowers do you think could explain the large TE burst in genomes that are 100 base pair and larger with 80% repeat content?
Okay. So what are the other superpowers? I think for retrotransposons, it's known that they're the ones, certainly in the plant genomes, that can increase copy number to thousands and tens of thousands of copies when they're transcribed. And I think their superpower really is that Mping doesn't have, is they insert into each other. So that's where you get these, when you have a genome that's 80% repetitive DNA, you have an 80% chance that you're going to insert into other repetitive DNA, which won't have an impact. And in fact, when you look at the different grass genomes, they're synthetic. You look at rice, you look at sorghum, you look at maize, and the genes are in largely similar positions when they are synthetic, but in maize, they're further apart. And that distance is what's driving them apart are the increased transposons. And it doesn't seem to matter because those blocks of transposons are essentially heterochromatin, and they're not accessible to, RNA polymerase or whatever. They're basically, Hugo Duner showed this, that the map units that separate like ADH and its nearest gene, you could have 100 kb that would be, one map, a fraction of a map unit, whereas within the gene, you would have more map units. So there's more map units where you have open chromatin than where you have heterochromatin. So it's a superpower is that they can keep inserting into each other.